All right, for my third and last video to try to sum up this information, we're going to look at the eukaryotic gene expression. So I see a lot of students struggling with like the terms and how they relate to each other, okay? So this is the structure of the gene, okay? Remember, it's just DNA. It's A's and T's and C's and G's. So when we color code stuff, we're just showing you that like this little section of code represents a control element, or this section of code represents the promoter, right? This section of code is what we say is the coding region. That's what codes for the polypeptide, right? So the enhancer, as I've written here, is the collection of the control elements, okay? This is going to be unique to each gene, okay? So the control elements is where specific transcription factors, which we've been calling activators, bind, okay? But activators are the same thing as specific transcription factors. That's the location where they bind, okay? Now, an enhancer can have, anywhere between 5 and 12 different control elements. Think of the control elements like letters in the alphabet, and the enhancer is the word. So different collections of letters create a different word, okay? You can mix and match letters, you can do longer words, you could do shorter words, you could do different letters. That's what makes these enhancers unique, okay? Now the promoter, you should know by now that that's where RNA polymerase binds. But in eukaryotes, our promoter regions have a TATA box, which is where RNA polymerase recognizes, okay? That's going to be basically like the start here sequence for where the gene is located. Notice again that eukaryotic genes contain introns and exons, okay? And they're really crazy long. All right, so that's just the structure. Now, the transcription initiation complex. Again, I'm getting so many questions about what this is. The word complex means like a structure made of multiple components, okay? It's the combined structure that includes your RNA polymerase, the specific activators for that particular gene, transcription factors, and mediator proteins. Now, those are more general things that are needed by every eukaryotic gene. All of these things come together to make the transcription initiation complex. I've got my textbook here to kind of show you. Okay, so you see here the transcription initiation complex. You've got RNA polymerase. Here are those little activators, right? Here are mediator proteins. Here are transcription factors. So use your textbook to help follow up with this stuff, okay? That's the complex. Right. Now, this is important. If any component is missing, transcription cannot happen. So if you're supposed to have 4 activators, but you only have 3, the complex can't form. That gene is going to be off, okay? There's no gene expression, that means no protein is going to be produced, right? So you have to have everything together to initiate transcription. Now, this is what I was talking about. So for gene expression to occur, think of this as a checklist. Are all of the activators present for every control element within the enhancer? Okay, if that's good. Has RNA polymerase attached to the promoter? Okay. Are all the mediator proteins and general transcription factors available? If you got check marks on all of these things, then you can initiate transcription. So the DNA will bend, bringing all of the components together to form the transcription initiation complex. Okay, so remember, your DNA is linear, but to bring these activators over to the promoter where RNA polymerase is, that's what this bending protein is doing. Bring that over. All right. So now you form the complex, But now go back and review protein synthesis. So now the gene can be transcribed. Remember, you're making pre-mRNA first. That's also called the primary transcript. After that's made, you got to modify that RNA. You got to remove the introns, you got to add the 5' GTP cap, you got to add the poly A tail, go back and review your spliceosomes, right? Then translation can occur. Read the mature mRNA and produce the polypeptide. So you need your ribosomes. You got to go back and think about codons and anticodons and tRNA and amino acids and how to use the codon chart and all of that stuff. All right? Okay, well, I hope these 3 videos have been helpful, but you got to study. So many details you got to study.